We may end up doing 3 chapters today. Not completely 3 chapters, but yeah. Touching 3 chapters today. Woo! Okay, so expression vectors. Expression vectors are going to be a class of plasmids that are optimized for production of large amounts of protein. The expression vectors will contain these things, which is an origin of replication, antibiotic resistance gene, promoter and terminator sequences. These will drive transcription, polylinker region, and transcriptional repressor and operator sequence to precisely control protein production. This right here is your a expression vector for a prokaryote. Okay? And again, note that these are typically plasmids. They are not the central run of DNA, right? So you could actually effectively put this in any prokaryote you wanted. Okay? You want to do it with heat and sulfur being the stuff it consumes, sure, throw it in a thermophile. Okay? Though it probably have a lot of issues itself because it wouldn't be optimized for that temperature. Anyways, right here is your transcription terminator, here's your operator, here's your transcription promoter. This is going to allow you to do the transcriptions. Here's your polylinker being many different linker types that you could use it with, the poly. Right here's your repressor, so you could repress this so it's not exhibiting at certain times. Here's your origin of replication, because it will have to replicate, otherwise you would have a species that grow out of it and would disappear. And then there's your ampicillin resistance. So the bacteriophages, these are going to be viruses that infect and replicate within bacteria. They alternate between 2 infection modes. There's the lytic pathway. That's the phage multiplies within the host and then lyses it once it's multiplied. The lysogenic pathway, this is the phage's DNA integrates into the host genome and lies dormant until activated. There's some really crazy human versions of these and plants versions of these that can lie dormant for decades. So this stuff actually gets pretty wild. Anyways, the bacteriophage lambda, there's the lambda, is a common cloning vector, meaning that it's a vector to do the cloning process through. Again, here's the phage right there. There's the DNA that's been introduced in the phage, or in normal natural circumstances, the phage's DNA, right? Right here we have the E. coli DNA and, of course, the bacterial cell. What's going to happen is this is then going to attack the cell, bind to it. It will then inject its DNA in. There's 2 different pathways from here on that diverge in their behavior. There's the lytic pathway. Lytic pathway being that it's going to lyse it, right? It's going to break your cell, okay? And so what will happen then is your progeny DNA will continuously replicate, make new phages inside, and then it will eventually lyse it when it fills up too much, okay? Everybody okay with that so far? Okay. Now, the other way is the lysogenic pathway. And the lysogenic pathway, that DNA that's integrated into your E. coli genome, it will then replicate with the E. coli genome, right? Over and over and over. And then eventually you'll have an activation. Boom. And then one day, all of a sudden, you replicate the progeny DNA, make a bunch of viruses around them, and they lyse the cells. It's a devastating sleeper agent attack. Okay? Okay. I know it's like Cold War happening in your cells. Well, not your cells, bacteria, but you get the idea. Mutant phages are going to be well suited for cloning. After restriction enzyme cleavage, the remaining DNA is going to be too little to be packaged into the lambda particle. The lambda DNA containing a suitably long DNA insert is large enough to be packaged. Modified viruses will enter a bacterial more easily than plasmids. And that's the main reason they're used instead of plasmids, is because they have these DNA segments that are not connected back around and are already being injected normally. If you just make a DNA segment with one of these lambda phages that has the gene you want expressed, you can have it do its normal attacking pathway, right? Whereas if we were to introduce the plasmids, I mean, this is round. It's not exactly like this is going to be the easiest thing to insert into a cell, right? These are typically have to be individually inserted into a cell. Okay. Yeah. All right, so the mutant lambda phage as a cloning vector. Right here we have the lambda DNA. This has the this is going to be the eco restriction enzyme right here. And then we'll have the removal of your middle section by the eco R1 digestion. And that's right here. You can see we're missing the middle now. We have a spliced foreign DNA, whatever we want to express. This would have the gene we want to express, or genes we're trying to have expressed. Oftentimes they do a lot of these with like, you've seen them like bioluminescent, duck, dog, cat, plant. Yeah. There's things that are not bioluminescent, by the way, but for some reason that's the thing. God, I don't know. Anyways, so what ends up happening is you're going to get two circumstances when you do this. There's going to be the two sides joined together. This will make a package that's too small to be packaged. So a fragment that's too small to be packaged. The other one is you have a splice of your foreign DNA that you're introducing that was also digested by the same restrictive enzyme. Note that this just has to be the same restrictive enzyme that can target the lambda phage DNA, right? Okay. Okay. By the way, there are designer lambda phage DNAs, in case you're wondering. Just like there are designer plasmids like we had just talked about. Okay. Anyways, this is going to do in vitro packaging of your recombinant molecule, and here we actually see that. This is the infective lambda virin. This is harboring your foreign DNA that we want to see replicated and actually expressed in something, right? Okay. Anybody have any questions? Fantastic. Note that because we have interrupted its DNA, most times what they've done is interrupted it in such a way that instead of doing this process right here, it will end up doing this over here, the lysogenic pathway. And then what will happen is it will then express that DNA sequence instead of replicating, okay, to make virion particles. Does that make sense? Yeah. Of course, if you don't disrupt that, and it still can make virion particles, you'd actually still get it sick, and then it would release the phages, and it could kill it. Yeah. Specific genes can be cloned from digests of genomic DNA. Your genomic library is going to be a large collection of DNA fragments. The probe is going to be an illegal nucleotide used to screen a genomic library for a gene of interest. The synthesized using knowledge of the protein sequence of the gene of interest. These are synthesized by the solid phase method and made radioactive with your 32P that we had talked about before. That is the phosphate with a weight of 32, right? Note that the reason that this is typically used as 32P, by the way, is because it's much easier to track when you don't have direct eyes-on contact, okay? So you can do it in situations where it has more thickness to it, okay? Because it's radioactive. Anybody have any questions so far? No? Okay. Okay. Do you understand the pieces that have gotten assembled in here? Okay. Okay. Yeah, it's using like 3 pieces from what we talked about before, right? Okay. Okay. Fantastic. So the probes can be generated from a target protein sequence. Peptide sequences will contain tryptophan and methionine. There's going to be preferred because these amino acids are going to be specified by a single codon, which makes this much more sure. You have a much larger, much less uncertainty in any measurements or what you're doing, right? If you have to depend on only one amino acid being correct, then it becomes a bigger deal, right? So again, these are your amino acid sequences. These are the potential oligonucleotide sequences. What this means is that if you have something like this, that's very easy to read and figure out the sequence for something like this, right? Something like this, it's harder to figure out the exact sequences. Not that matters for the protein expression, but you may not actually get the correct sequence from that. Okay. So this is the preparation of genomic libraries. Step one is going to be to fragment a sample containing many copies of the total genomic DNA. This right here being that genomic DNA right there. Step two is separate the fragments via gel electrophoresis. And so we have separated them. Typically, this can be done through shearing, or it can be done through digestion with restrictive enzymes. Restriction enzymes, excuse me. We would then join these two lambda DNA pieces. And so we see that we've assembled them with the two outer sides of the lambda DNA pieces. Remember, we cut out the inside, and then we assemble in our previous figure. Okay. And so what this is going to give us is it's going to give us different packages that we could then have the lambda virons incorporate. So we'll have the in vitro packaging happen next. We'll attach synthetic linkers and form cohesive ends. Step four is then going to be to insert the fragments into a vector that we prepared with the same cohesive ends. This is that part right here, by the way. Step five is going to be insert the vector into E. coli and amplify. So this is the part where we're going to insert the vector into E. coli and amplify right here. Okay? Okay, and that's the lambda viron. It'll have our foreign DNA fragments. They're going to attack the E. coli. They'll inject this DNA. That DNA will get incorporated into the E. coli's DNA, right? Okay. Fantastic. Those infective E. coli will duplicate, and you can think of it being essentially this pathway right here. Okay? That's essentially what we've done, right? And we've disabled this DNA from being able to do the progeny. Okay? Because we cut it off. Mm-hmm.
So are the latex squares on the image colors, is that the synthetic linkers?
Okay, so the synthetic linkers would be these parts. Yeah. And that happens because oftentimes, remember we synthesized this? So we basically reverse transcribed this and then made this. And it's not going to have nice E. coli attack sites, right? So we have to build out our own linkers that will link to those EcoR1. Okay? If you're not using EcoR1, of course, you would, build it out for whatever you're using. Does that make more sense? Okay. Yeah. That's the synthetic linkers. That's why we have to do that. Then once we have the synthetic linkers, we'll have a fragment that's foreign DNA that will match our restriction that we'll use on the lambda DNA. Okay?
So are the light, what are the light?
The lightly colored yellowish pieces are these. The lambda DNA sides. Okay? And then the colorful ones are these ones right here, our inserts that we're putting into them. In that case, it's different synthetic ones that we had to build out linkers on the sides, which are that stuff right there. That make more sense?
Yeah.
Oh, no, I'm glad you asked. Thank you. Honestly. Like, you guys engaging and asking is actually quite helpful. Yeah. And, that's the whole reason for a class, right? That's why you have me. All right. So, your screening and genomic library for your specific DNA, of course. We have the rapid screening. This can be accomplished by DNA hybridization with a 32P labeled probe. The autoradiography will reveal the position of your spots that harbor the recombinant DNA. So right here we have the plaques. This will be on a master plate. We're then going to apply our nitrocellulose. This is going to then essentially blot our plaques, okay? It's the same process we did in blotting. Okay, So then we have the nitrocellulose replica of the master plate. This is one of the reasons why that's called the master plate, right? Okay. You don't do it on the master plate because then you'd be out your master plate. In case you're wondering. Okay. The next thing we're going to do is we're going to put an NaOH and a 32P labeled probe. By the way, what is the NaOH going to do? It's going to digest a bunch of this stuff, right? It'll actually break it up. Okay? So if you did this to your master plate, it's dead. That plate's done for. Okay? Any cells on it, anything on it is probably going to be partially destroyed. So the next thing that's going to happen is we will then have this labeled probe. It will attach to a specific site that it matches up with, right? And when it attaches to a specific site, that actually tells us that this one right here, this one detected as being radioactive. And so that means that's the one, the incorporated thing we're looking for, isn't it? Okay. Okay, and then we do an X-ray film, and that's going to give us an autoradiographic probe labeled nitrocellulose. Does anybody know what nitrocellulose is? You know the old film, Kodak film cameras? That's that. Okay? And so it being exposed to X-rays gives it, right? Or it being exposed to radioactive material causes it to blot, right? To develop a color, like film. Okay. Yeah, neat, huh? All right, so the complementary DNA prepared from an mRNA, these can be expressed in host cells. By the way, in case you're wondering, that's like the main thing that Kodak does now, is it just makes nitrocellulose stuff and chemicals. It almost has nothing to do with film or cameras at all anymore, in case anybody was wondering. So this is going to be the complementary DNA prepared from the mRNA. This can be expressed in host cells. Genes with introns cannot be expressed by bacteria, of course. So if you're trying to replicate something from a eukaryote, you actually have to get the one that's expressed as the mRNA, right? Because the prokaryotes are not going to take the exons and just express those. They would express the exons with the introns, dividing them, and so you won't actually get the protein, right? Okay. Now the complementary DNA, this is the cDNA. This has a designation. Everybody clear with that cDNA, complementary? Okay. This is going to be the double-stranded DNA molecule we generate using an mRNA template by reverse transcriptase, which is a retroviral enzyme. And so we basically feed something that is the mRNA that would come out of the cell and do the translation right later on. We feed that into the mechanisms of a virus that has RNA as its base. Okay? And then its reverse transcriptase is going to reverse that into DNA, right? And then we can incorporate that DNA where we want it. Does that make sense? Okay. Does it make sense why we do it? Okay. Okay. Yeah, because the introns, right? We need them to be out of it. Because the damn prokaryotes don't do that. The cDNA library is going to be a collection of cDNA for all mRNA that a cell contains. The cDNA is going to be inserted into vectors that are inserted into bacteria, so then you can get an E. coli that makes human insulin. Okay? Fantastic. This right here is the formation of your double-stranded cDNA. And so here we actually see the mRNA. Again, that's the 5', 3', that's the poly A tail right there. Everybody okay with that so far? Okay, we'll attach the oligo T primer, literally repeated T's, that will match up with the repeated A's, right? And then we're going to put it with reverse transcriptase and supply of deoxy NTPs, right? Materials. And then this is going to make the cDNA from the 5' to the 3' along that. The next thing we'll do is we're going to have an alkali digestion of the mRNA template. What's then going to happen is we'll attach another oligo, in this case a DG template, primer region. This is going to leave a repeated T's on one side, repeated G's on the other. Okay? And so now we have buffer regions where we can successfully attach stuff to both sides. Okay? Note that our regions here are different because we don't want to have the same primers attached to the same places, right? Yeah. Okay, so the illegal C primer is going to be attached. That's going to give us our primer. We'll then put the reverse transcriptase again and more supplies of DNTPs. This is then going to replicate the DNA and give us the other side. And now we have both sides. Okay? Anybody have any questions on that? Fantastic. Okay. Everybody understands why we have to put the repeated G units here? And this isn't just good up here? We need a place to attach a primer so we can synthesize the correct direction again. Okay? And it can't be the same primer or else we would try to synthesize this way. And you'd have a bunch of failures. Yeah. Yeah. Okay, so expression cloning. This is going to enable the production of the corresponding protein of interest. The expression cloning is going to be a technique by which the cDNA libraries are prepared in expression vectors and screened for the capacity to direct synthesis of a protein. So right here we actually see the graphic representation of this, which I think is clearer, but you need definitions. Okay? So you got your definitions now. Right here we have the bacterial promoter site. Right here is going to be the eukaryotic cDNA insert. Remember, we just made that right here. That's the cDNA insert. It's the expression of a gene as could be read by a bacteria, right? From the mRNA of a eukaryote. Okay? It only is going to include what's? Exons, right? And it's actually important, they also have to be arranged in the order they should be. Yeah. So if it would have gotten shuffling happening, it needs to be the mRNA that would go off to the ribosome. Okay? So it needs to be the actual instructions. All right. So this is going to transform your E. coli, of course, once we put that expression vector plasmid into it. And so you'll have a colony that will start producing the protein of interest. These bacterial colonies will be grown on agar plates. You'll transfer colonies to a replica plate so you can replicate the colony of interest, right? The colony that produces the protein of interest. You'll transfer the proteins to a nitrocellulose sheet. You'll add radio-labeled antibody specific for that protein of interest. This is then going to develop dark spots using your autoradiography, just like we talked about before. That's called an autoradiogram, in case you're curious. It doesn't really show up much, but it does show up every now and then. Okay. Yeah, which is the right name for the autoradiography when it is 1 gram, right? One reading. Okay. Anyways, does anybody have any questions about this? The last part's the same thing as we talked about before, right? Yeah. And everybody understands why you have to isolate and grow your colony? If you don't isolate and grow just the colony you want, what will happen is it may get taken over by the others. Because it is making a protein that's not really necessary, right? Evolution-wise, that wasn't a great move. All right. So the bacteria can infected with proinsulin, cDNA. This can express the eukaryotic protein. This is actually what I mentioned before as an example, in case you're curious, because it's probably one of the most important examples of this, in fact. Of course, here we have a pancreas, right? Normally it would express the mammalian proinsulin mRNA. And in a normal pancreas, this is then going to go on to translation, will make the protein. It will then go into the bloodstream, right? And then we have proinsulin. It gets activated. Bam. Now we have insulin that tells us, hey, take this sugar and fix it. Okay? Okay. But what we're going to do here is we're going to make a cDNA. So we're going to put this proinsulin mRNA from a mammal. We're going to put it with reverse transcriptase. Same process as we looked at that previous diagram. We'll get the proinsulin cDNA. We're then going to join that to a plasmid. This is the recombinant plasmid then. Again, see here the gene for the proinsulin. And then we're going to infect E. coli with this gene on a plasmid again. This is then going to give us the mRNA for the proinsulin, and eventually the proinsulin is going to end up being made in these cells, right? Okay. And so we'll now have E. coli, which grows very fast and with very little nutrients added to them, right? That can rapidly make a lot of proinsulin, right? Oh, and so then we can take these, we grow them like little algae pond things, okay? Little vials essentially. And then we grind them down, extract the insulin, proinsulin, and then we can use it on people, and now they have proinsulin, right? Whereas before they did not. Okay, anybody have any question? Fantastic. So proteins with new functions, this can be created through direct changes in your DNA. Recombinant DNA techniques actually allow for the introduction of specific mutations in vitro. The three main classes of mutations are going to be deletions, insertions, and substitutions. You guys have all probably heard of that, haven't you? In genetics. Okay, fantastic. The site-directed mutagenesis. So your site-directed mutagenesis is going to allow the generation of mutant proteins with a single amino acid substitution. Your point mutation is going to be the mutation where only one base is changed. Makes a lot of sense in terms of terminology, hopefully. The site-directed mutagenesis is going to use mismatched primers in a PCR. And so here we have the base that's to be altered. We'll have the parental plasmid here. This is methylated DNA. So we'll then separate the plasmid strands. We'll add a mutagenic PCR primers. And so here we see that we've added a primer, and see how one of them is mismatched. Yeah, that's not right, is it? Okay. But because all the others are matched, it binds pretty well. Okay? It'll be hard for that to come off. And so, again, we have the other side here. Everybody okay with that so far? We're then going to run the PCR, and because in the PCR we only have one type of replication going on, right? We don't have the full set of polymerases, do we? And so what happens with that polymerase is it only starts after our primer, doesn't it? Okay? And because our primer is the one with the mismatch, that mismatch never gets cut out. So it always stays. And so we can incorporate the mismatch ourselves. It'll replicate the rest of the DNA through the normal PCR process, but our mismatched primers will always maintain their mismatches. Okay? Does anybody have any questions about that part? Okay, so then you're going to digest the methylated DNA with DPNL. This will then transform into bacteria. And so right here we now have the mutated plasmid. Again, the parental ones were the methylated DNA. So we've gotten rid of the methylated DNA, the parental ones, the ones that did not have mismatches. And so we're just left with the mismatched copies. Okay? Okay, anybody have any questions about that? Fantastic. Okay, cassette mutagenesis. I promise you it does look like cassettes. Have you guys seen cassettes? Okay. Okay. I mean, they were gone before you were born, so I don't blame you if you haven't. So cassette mutagenesis, this allows the introduction of a variety of mutations to a gene of interest. The cassette is going to be a synthetic double-stranded oligonucleotide that will carry the genetic alterations of interest. This is prepared with cohesive ends that are going to be complementary to the ends of the cut plasmid. It's going to be inserted into the gene of interest. So right here we actually have the plasmid with the original gene. We're going to select cleavage sites on both sides that'll incorporate the gene that we're interested in. Okay? And so we then cut this with restriction enzymes. We can use 1 and 2 if we would like to get the exact one we need. Okay? Okay. Notice this does not have matched ends. See that? This is different than previous. Previous we'd always used matched ends. So we used 1 restriction enzyme to cut both ends. Okay? This one does not. So restriction enzyme 1, 2, and then we actually cut this up into more fragments, 3, 4, 5, if we want. Anyways, the important one is this one right here, the blue one. We're then going to purify the large fragment. So we purify that. We're then going to add the new cassette ligate. We'll purify your large circular plasmid, so we only have the one with the new gene. Okay? Okay. And again, in this case, one of the big differences here is this has directionality, right? Because we have different sticky ends. We have sticky end 1 that corresponds to 1 restriction enzyme, right? And we have sticky end 2, which corresponds to a second restriction enzyme, right? And so in this case, we have directionality, okay? Much like a cassette has a forward and backwards direction. Okay? Does everybody see the cassette, by the way? Yeah? Yeah. The cassettes had a little spot that rotates around, right? Okay. There we go. So again, mutagenesis by PCR, this is going to use PCR primers to introduce insertions, deletions, and substitutions. Again, you know that primer, we can pretty much just put whatever we want in PCR, right? Because the primer doesn't ever gets checked, okay? So you may have noticed that we could have done anything for that primer we basically wanted, right? And so the inverse PCR, this is going to introduce deletions into plasmid DNA. And right here we see what we're looking at. And so what we see here is this is that gray region. We're going to delete that. So again, we're going to open this up by cutting out that gray region. We're going to purify that. We're then going to amplify that by PCR. And then what's going to happen is we're going to take these 2 sticky ends on each side of that gray region, and we're going to rejoin them. Okay? By the way, anybody tell me why we don't do the PCR while this is still round? In PCR, are we doing the normal polymerase reaction with helicase and all that? We aren't, are we? We're not using the normal helicase, okay? Meaning that we need to melt these. We actually detach the full strands completely, don't we? And then replicate the 2 strands individually, okay? And so if it's still contained together, it's actually going to be very difficult to detach those, isn't it? Okay? It's actually impossible to detach them, as we'll discover more as we go on. But designer genes. No, this is not genes that you buy at the supermarket. So designer genes, these are going to be novel proteins. These can be created by splicing together gene segments that encode domains that are not associated in nature. Actually, I just lied to you. These literally are genes that you can buy at the supermarket. In your food. That's how you can make certain GMOs with like resistance to pesticides, by the way. So you can buy it at the supermarket. The immunotoxins, these are going to be chimeric proteins that are formed by joining a gene for an antibody to a gene encoding a toxic protein. What these do is they're going to kill any cells that are recognized by the antibody. These are actually being evaluated as anti-cancer agents. One of the big reasons they're being evaluated as anti-cancer agents is because cancers tend to take in things that normal cells would not take in. Okay? And so you can actually target them quite nicely. And then send a death signal, which is what cancer doesn't do, right? It doesn't take on a death signal. Synthetic vaccines, these are non-infectious proteins of viruses produced by recombinant DNA methods. These are theoretically safer than conventional Vaccines prepared by inactivating pathogenic viruses, because you will have no virus in it. However, there's been some concerns raised in the recent past because of the entire mRNA debacle with COVID, right? And they have found that full DNA actually does sometimes get into these things, and more than you'd think. So it's been a bit of a controversy, just to warn you, if you decide to go in that field. So, not that it is an amazingly cool field, but just so you know. The complete genomes have been sequenced and analyzed. The genomes of organisms ranging from bacteria to multicellular eukaryotes have been fully sequenced. This right here is some different examples of sequencing and what they've found from that. Anyways, this is a trebly complex piece of information that you actually get fairly little out of, but it's pretty cool, okay? A lot of work went into it, to be fair. Anyways. So the sequence of the human genome has been completed. It's been completed for quite a while, like 15 years now. This is comprised of approximately 3 billion base pairs of DNA. These are distributed among the 24 chromosomes. That is 23,000-ish genes. This number actually goes up and down every now and then because new things are discovered about exactly how that encoding works. Okay? Remember that you can have one stretch of DNA that codes for multiple genes, right? And some of the stretches of DNA don't encode for anything. And some of the stretches we thought encoded for nothing do encode for stuff. So new stuff's being found out all the time. Here's some different examples of these and the chapters they exist in, though we haven't gotten to a number of them. So for example, right here, we have the 3-hydroxy 3-methylglutarate coenzyme A reductase. That's the chapters we'll get it on. Anyways, here's your chromosomes. Yay. Okay. Anybody have any questions? Fantastic. Pseudogenes. These are going to be formally functional genes that have accumulated mutations such that they no longer produce proteins. More than half of the genomic regions that correspond to olfactory receptors are actually pseudogenes. This is one of the reasons why humans are so bad at smelling when organisms that were very close to us are much better. You guys okay?
No, I'm like this one.
Okay.
Sorry, my heater.
No, that's fine. Okay, so the next generation sequencing methods, these are going to be sequencing methods that are coming up, and some of them have actually started becoming more widespread. So they're going to enable rapid determination of a complete genome sequence. The next generation sequencing, NGS, this is going to be a family of technologies that makes sequencing of whole genomes rapid and inexpensive. Right now, does anybody have any idea about how much money it takes to get a full sequence for you? You can get partial sequences for like $1,000. You can get full sequences, or typically somewhere in the 10 to 20-ish thousand. Though, considering medical procedures, that is not a lot, actually. Like, keep in mind, this could cause you to not go $1 million in medical treatments for cancer, right? So, actually not that expensive even now. And that's because these have already started kind of showing up, okay? They're not perfect now, but they're starting to show. So NGS is going to rely on advances in handling small amounts of liquids, of course, high-resolution optics, and computing power. These allow you to do more and more with less and less, right? All the methods are going to be highly parallel. Between 1 million and 1 billion DNA fragments will be sequenced, are going to acquired in a single experiment. I mean, think of that number, right? You're doing 1 to million to 1 billion DNA fragments in one go. Okay? Imagine if I tried to make you do that in lab. with the methods we use. Do a million reactions today. Okay, yeah. And this is why, basically, it's a lot of automation with computer power going on here. And the automation can handle these small liquid amounts to run, a ton of experiments all right next to each other. Yeah. So the individual DNA fragments are going to be amplified by PCR. This is going to be done on a solid support. The fragments will serve as templates for the DNA polymerase. The addition of the nucleotide triphosphates is converted to a signal that will allow for the identification of the added nucleotide. Some common examples are NGSs are going to be the reversible terminator method. This will use a method akin to the Sanger dideoxy sequencing. Remember that from previous? Okay, it's very similar. It's just we can now detect right as we're doing it, rather than having to sequence the end result. Okay, so we're cutting out a bunch of steps. Pyrosequencing, this is going to be a method in which nucleotide addition is detected by a flash of light. And so you can monitor the additions when you wash these over the solids. You caught that these were solid support, right? They're a solid phase. So you can wash your nucleotides over them and introduce the nucleotides sequentially whenever you want. Okay? And so you can wash a nucleotide over, flash a light. That's the one that was incorporated, right? And so now we've sequenced that one. We just keep doing that really fast. Okay. The ion semiconductor sequencing, this is going to be a method in which a released proton is detected upon nucleotide addition. Again, the big thing about NGS is it's that our photovoltaics, our ability to detect when things happen, our computers are fast enough to detect those happening really fast. And instead of getting the end result and then figuring out what that end result is, which requires 10 extra steps, we're making it and detecting it at the same time. Okay? So again, here's the pyrosequencing reactions in more detail. The DNTPs. Still too hot. The DNTPs are going to be added one at a time to the sequencing template. If the DNTPs are added is incorporated, the release pyrophosphate is then going to be coupled to the production of light. This flash of light then will hit a detector. This is going to occur by the sequential addition action of ATP sulfurylase and luciferase. The emitted light can be measured. This will reveal the base identity at that position. This is the actual reaction, by the way. And so we have these free phosphoryl groups, adenyl sulfate, in the presence of ATP sulfurylase. This is going to react. It will give us ATP and sulfate. The ATP and luciferin is going to react in the presence of the enzyme luciferase. This will give us the oxyluciferin, and a light will flash. Well, light will evolve. Okay? And so we're then detecting the light, recording which of the NTPs was going through at that time, and that gives us which one attached, right? After the nucleotide addition, the remaining deoxynucleotide and ATP are going to be removed by aperase. That's this right here. We need to reset it each time so we don't continue having reactions. The next nucleotide will then be added. The steps will be repeated over and over and over, adding one at a time and detecting if that actually did add or not. Okay. The dispensation sequence is the defined order that the nucleotides are going to be added in. This is important to actually understand what you'll be adding when, because if you mislabel, then you will have the wrong sequencing, right? Okay. Anybody have any questions about that? Did that make sense? Okay. Again, that pyrosequencing is an example of NGS. This is actually the diagram that shows that. And as you can see again here, we have the solid template strand that we're adding to. we have to put a primer on it. And so then what's going to happen is we will then add an ATP in this case. So we're getting the D-adenosine. It will have no reaction because the next one here is not an A, is it? Well, T at matching to an A. Okay? And so we then put aperase that will remove any additional ATP that's still on there. And then we'll add the next one. In this case, this is a D-CTP. This will actually add because C matches G, right? And so that will release the PPI. This will go through a reaction where it goes to ATP and then light. And so now light photons start hitting our detector and it goes, hey, we put CTP in there and it added, okay? And then bam, we get a signal. Okay. And again, we reset our system with aperase. This removes any of the excess CTP or ATP. And then we keep doing that over and over and over. Okay? Cycling through our 4 different bases each time. Does that make sense? Okay. Fantastic. And this is actually what we would get out of this. And so see that we have ACGT, 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 right? And we would get a signal from C in this case, then we got a T, we got another T, see how it's a double signal? Okay. So it added twice, and so we get twice the amount of light. Okay. And then we got an A, and then a G, T. Everybody good? Fantastic. Yeah, pretty cool, huh? It's like we're summing everything up we've learned. Yeah. Okay, so we can use different approaches to measuring nucleotide incorporation. They all have some similarities though. So we'll have a primer, we'll be adding the next ones to it. In the case of pyrosequencing, the PPI does that reaction which creates that flash of light, right? In the case of ion semiconductor sequencing, we release a H+. This causes a charge difference, right? And so our semiconductor actually will read those ionic charges coming off as they happen. And then this is the reversible terminating sequence, which has that reversing termination right there. The comparative genomics has become a powerful research field or research tool in its research fields. 99% of the human genes have counterparts in mouse and rat genomes that have been reassorted among chromosomes. In fact, if we were just to look at how much DNA we share, if we were just to take, we have this sequence, do they have that sequence? Most animals actually share a tremendous amount of DNA. We're all pretty much similar, okay? Of the animals we would say we have anything to do with, typically we share something like 60 to 70% of our DNA, okay? The difference between a chimpanzee and a human is about 5%, okay? Okay, so this is your human chromosome, these are your mouse chromosomes, and as you can see here, if you look really closely, it's actually mostly just rearrangement among the different chromosomes of these gene runs, these fragments. Anybody have any questions? Okay, so eukaryotic genes, these can be quantitated and manipulated with considerable precision. Our gene expression levels can be measured by measuring mRNA quantities. These are going to vary from cell type to cell type because different expressions will happen in different cell types, right? The change in response to physiological circumstances will also change how our DNA is expressed. This does not always correlate with protein expression levels. In fact, we can have situations where we are correctly coding for the protein, the correct amount of mRNA, not getting the correct amount of protein out, okay? You probably already kind of knew some of this. Like you may have known that stress reactions can change how your DNA presents. anyways, it's kind of become a famous field. The quantitative PCR, so quantitative PCR or qPCR or real-time PCR, it has 3 different names, congratulations on that one, is going to be a technique to determine the quantity of individual mRNA transcriptions. The SYBR green 1, this is a dye that fluoresces brightly when bound to double-strand DNA. The cycle number at which the fluorescent signal exceeds a defined threshold, that defined threshold is C of T, literally meaning cycle, and T meaning the threshold. Okay? This is going to be inversely proportional to the number of copies of the original template. So the more copies of the original template, the faster you get to C of T, right? Because you'll build up that population. It's like saying, if I start with $5, if I start with $1 million, who's going to get to 2 million faster if we invest? Probably the 1 million, right? Yeah, and that's what we're looking at here. Okay, and that's this is monitored through fluorescence. That's how we can monitor the concentration, just like your normal Beer's law fluorescence concentrations that we had talked about before. And that's what we're seeing right here. Anybody having questions? Okay. Again, this right here would be these arranged. So we got the C of T's for these different fragments right here. And then we arranged them in a graph. And this shows you the starting quantities if we use that relationship. Okay. And so, for example, the blue one right here, this started with the least, because we needed the most cycles. Okay. By the way, everybody notice this is like a sigmoidal relationship right here. Okay. See the blues are over here and then they go up here. We start detecting them. Okay. DNA microarrays. So these are going to be transcriptome, the pattern and level of the expression of all genes in a particular cell or tissue. The DNA microarray is going to be used to determine the expression pattern of a large number of genes simultaneously. The gene-stranded oligonucleotides are going to correspondingly to coding regions of the genome are going to be affixed to a solid support. Man, this needs to cool down. I'm dying here. Fluorescently labeled cDNA, remember this is the complementary DNA that we can get from the mRNA that we talked about before. Okay, this is going to be hybridized to the microarray to reveal the gene expression levels. And again, we're looking at how much that DNA is expressed in that specific cell under those specific conditions, right? Remember, your DNA is actually the same chromosomes all over, right? But it is not expressed the same everywhere, is it? Or in every situation. So again, here's our DNA microarrays. We're measuring, in this case, gene expression changes in the tumor. So here we have a normal cell, here we have a tumor cell. So the first thing we're going to do is we're going to isolate our mRNA, and here we have our mRNA isolated. The next thing we're going to do is we're going to prepare the cDNA in the presence of fluorescent DNTPs, excuse me. I don't have any water. Oh, yeah, my tea needs to cool. So again, we got our cDNAs like we talked about before. They're fluorescently labeled so we can see them through fluorescence, right? The next thing we're going to do is we're going to separate the cDNA into single strands. We'll then add our microarray. The fluorescent strands will anneal to the complementary sequences along this sheet here. We will then wash the unhybridized cDNA strands off of this sheet. So we just have the ones that have hybridized. The red spots will indicate the increased expression in tumor related to the normal. And so we right here, we see the fluorescently labeled red ones right there expressing. Everybody okay with that so far? Fantastic. Green spots are going to be a decrease in tumor relative to normal because the green will start showing through, right? Yellow spots are going to be high expression in both samples, so we get both. And then black spots will be, well, we didn't get anything fluorescent, right? So, yeah. Low expression in both. Anybody have any questions about those? Yeah. Essentially, we're getting the DNA fragments to stick certain spots, and this just allows us to visually indicate which of those has higher expression. Okay. This is actually what that looks like. I know that we give you this, but that's not what anything ever looks like, right? That's a beautiful diagram that somebody, made. That's made up. This is what that actually looks like. It's a little bit more complicated to look at, but not too bad, right? Can everybody read that okay? Yeah. So for example, this right here means what? The black spot. Yeah. Low. Yeah, we're not getting basically any expression for either. How about this really bright red? Exactly, you got it. And these are just different genes that we've put the arrays that will stick to them. And so we would read over here, this would be a specific gene. Okay. And then these are different tumors that have been let go through here. And so we could see tumor, whatever, brain tumor, pituitary tumor. You get the idea. Okay. And what genes are being expressed. Okay. And note, by the way, again, right here, for example, we have a lot of like expression that's this normal cell expression is expressing more of, right? Okay. So the RNA sequencing, the qPCR and microarray are going to require transcript sequences to be known prior to the experiment. The RNA sequence is going to use to detect the presence and quantitation of virtually all RNA molecules in a cell under specific conditions. It's more sensitive than the microarrays. It can be used on individual cells, the single-stranded RNA sequence, or SC, RNA-seq. So these are the steps in doing this. We have step 1, which is going to be isolate the RNA from cells or tissues of interest. You can actually run this on very large samples, which is kind of interesting, right? Yeah. But you'll have a bit more fuzziness, if you will, because remember, cells will have slight differences sometimes. Okay? It's real world, like your spinach, right? Okay. Step 2, this is select for RNA by filtering for sequences containing poly-A tails. Remember that our RNA is going to have the poly-A tails, right? So if we stick things with poly-A tails on them, we're going to get the RNA. Okay? Step 3 is going to be to synthesize the cDNA using reverse transcriptase. Step 4, that's the same way as we talked about before, where we sequenced the cDNA from a poly-A tail. Remember where we primed it at the poly-A tail, replicated it? Okay. Step 4 is going to be to sequence the cDNA molecules using an NGS method. You could use the older methods, they'd just be really, really slow, right? So the NGS method, much faster. Step 5 is to use a computational algorithms to assemble the sequencing data. Typically what you do with these is you would have a bunch of different cDNAs that you're going to sequence, and then you would assemble those because they're going to have some spots that match up, and you can assemble them into more complete genes. You may not get the full thing and have to do a few more experiments, but you'll get pretty close. The new genes can be then inserted into eukaryotic cells that can be efficiently expressed. The eukaryotic host cells are going to contain enzymes to carry out post-translational modifications. The recombinant DNA molecules can be transformed into animal cells by a variety of methods. Some of the different ways are transfection, The process by which foreign DNA will be precipitated by calcium phosphate can be taken up. Microinjection of DNA into cells. You've seen this. You've seen Jurassic Park, right? You've seen it. yeah. Transduction. This can be the process by which viral vectors introduce new genes into animal cells. And so remember that if we're talking about eukaryotic DNA, we could express that DNA, have it replicated many times, and do a lot of useful stuff with the prokaryotes, but we'll never get the actual expression of the DNA, right? For eukaryotic DNA in a prokaryote, unless we make a cDNA first. Okay? Huh? What's up? The microinjection. Yeah. This is not that you actually have to do it like Jurassic Park. Yeah, keep in mind that's 1990s, right? Yeah, and it's a movie. It's not a real science lab. So it's pretty convincing looking. You can do much more careful injections than that. Yeah, there's a lot of stuff that's going on now. But anyways, this would be the most Jurassic Park-like. Okay. Anyways, everybody understands that if you feed a prokaryote a eukaryotic DNA that has not had its exons only put in, or does not have those arranged properly, that they're not going to be able to do anything with it, right? Yeah, they're not going to get the actually expressed proteins that you get in a eukaryote. So you have to feed it into a eukaryote to get those expressed properly. Okay? Okay, so this is the microinjection, by the way. This is what it actually looks like nowadays, usually. You can actually do this under a microscope with just your hands, but it is really freaking difficult. So normally this is a robot of some sort. You'd have a holding pipette that literally just does a little bit of suction on your fertilized mouse egg. And then you insert this, and then you actually need to get it into the, see how it has to enter through? And then you're going to inject the solution with the DNA. Does that mean that sometimes you get more than one? Yes. Yes, you're almost never just going to get one of the DNA fragments in there. You just have to basically scatter them, shoot it into a bunch of these, and then you fertilize like 100 of them and see if any of them take. Okay? And that's actually why, if you ever pay attention to the these labs that do this with mice that inject DNA fragments or clone, they'll be doing like hundreds of fertilized eggs, and then they'll do them like batches of a dozen or two, and then most of them don't take. Okay. Anybody ever heard of like the 12 kids? I had 12 kids, and I was doing a birth treatment with my doctor where they fertilized. No? Okay. Look it up on the internet. There's some really weird stories where people have like 12 kids at once. And the reason that happens is because when you do fertilization like this, same method, they take the DNA from the father, who does not have effective sperm usually, and then they take the egg from a mother or someone else. They insert their DNA. And then they transplant those eggs into the uterus of the mother, right? But they don't transplant one. They transplant like a dozen and see if they take. And typically what happens is the majority of them die, and you're actually lucky if one takes. Yeah. Because again, it's a scatter shot, right? Yeah, that does not happen a lot. Okay, but you can look it up. It does happen. I mean, most of the time this works fine. Okay? And if you have extras, what they usually do is they go in, they'll see them start attaching. Most of them don't even attach. And if you start having a bunch, you can just take like the morning after pill, essentially. Yeah. And you can terminate that one. It will cost you money. This stuff costs like $10,000, $15,000 to go. But yeah, anyways. So the transgenic animals harbor and express genes introduced into their germlines. The transgenic mice, for example, are mature mice that will have incorporated a foreign gene into their genome. They generated via microinjection of plasmids that harbor that foreign gene into the fertilized mouse eggs. There's actually is designer mice that exhibit human proteins, so that you can test interactions with human proteins in them. Okay? They're used to explore the role of gene in development, growth, behavior. They can be used as models for disease processes. These are that same type of mice that we use a lot of models for, like infections with diseases and stuff. Okay? Gene disruption and genome editing are going to provide clues to gene function and opportunities for new therapies. The gene disruption or gene knockout being the far more common, because it's fun to say, I bet. But anyways, gene disruption as well. The technique by which a foreign DNA fragment is inserted into a genome by homologous recombination. This is the graphic right here for it. And so we have a flanking sequences, 5' and 3'. Here we have the mutated gene with foreign DNA. And so this right here is then going to be the targeted gene with genomic data. And homologous recombination is going to involve swapping these out. So see, we had the blue one in the sequence, right? And what we'll do is we'll have the mutated gene. This could be one that doesn't function. This could be one that does not, the enzyme functions less well than it should, like a normal, like diabetes gene, right? That'd be an example of this. And so then we put the mutated target gene into the sequence. Everybody okay with that so far? Okay. The homologous meaning it's same size, same position, same switch out, right? We just have a mutated one. Okay, this is the consequences of gene disruption. This is your wild type here. This is myogenin knockout. So what we see here is that we have these muscle-forming progenitor cells. They'll form our myoblasts. These should assemble into myogenin. Well, with myogenin, they should assemble into myotubes, which would give you mature skeletal muscle. That's that wild type. See the nice-looking skeletal muscle? Myogenin knockout. Not nice-looking skeleton muscle, is it? Don't even look like fibers almost. Yeah, very broken apart and obviously not functional. Okay? And you can see exactly why a lack of myogenin would cause that if you do this. You can start testing various hypotheses and stuff, right? So genome editing, this is going to be a technique allowing for highly specific modification of genomic DNA. This is going to rely on the introduction of double-stranded breaks within genomic DNA. Non-homologous end joining, which is NHEJ. God, I am just. This is finally cold. No, stop. God, I need to get worse heating things. I didn't filter any water today, so which is a mistake. Okay, so again, these are NHEJ. They're going to be error-prone repair processes when no template is provided. They're going to often introduce insertions and deletions that result in a gene knockout. Homology-directed repair, which is HDR. This is going to be a repair process that uses the donor template to introduce changes directly into the genomic sequence. This is actually your genome editing. Again, and it starts with the introduction of your double-stranded breaks in your host DNA. And so right here we have our double-stranded break. See that introduction of your double-stranded break? Let me roll them apart. Good. This would be the non-homologous end joining. And so here you see that we have not introduced these and a homologous fashion, have we? Yeah. It's not a homolog, is another way of saying that, right? And so what's happened is we have an insertion or deletion that has occurred that is then going to knock out that gene that's being expressed here, right? It's no longer going to be a functional gene. This right here would be a donor template DNA attaching. This one would be a homology directed repair, HDR. And so again, this is going to have a homolog for these portions that's going to replace it. It will introduce that new portion right here, which is the homolog directed repair. Okay. Does anybody have any questions? Cool. Zinc finger nucleases, ZFN. The zinc finger nucleases are going to be engineered nucleases that can target any region of your genome. These are formed by combining the focal nuclease domain with the DNA binding domain that contains a series of zinc finger domains. Preferred DNA binding sequences can be altered by changing 4 residues with each finger. Transcription activator, like effector nucleases, TALENs, TALENs. Transcription activated, blah, 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 you got it. Engineered nucleases that can target a vast number of targeted sequences with high specificity. They're actually formed by combining the focal nuclease domain with an array of tail. Again, that's the transcript activator like effector. We just didn't include the nucleases, okay? So tail repeats. The recognition sequence can be altered by mutation of residues 12 and 13 with an array of repeated changes. Right here we can actually see that, so we can get a visual. The tail repeats are going to recognize individual bases in your DNA. And so right here we have the array of tail repeats. You can see that they'll match up to a specific sequence of your DNA there. And that's what it looks like right there. Okay? And see how they're attaching to the phosphate portion? All right, very good. Why are they going in here? What the hell? Why did I skip there? This is the wide, narrow. See that in your DNA? And so these are the parts that I can get access to the base, right? Yeah. I can't get access to the base in here. I would only hit the backbone. Okay? And so it literally follows along the wide, interacting with your bases there. Okay, so 2ZFN TALEN constructs introduce double-stranded breaks. So we have the ZFN TALEN number 1 and then a focal nucleus domain right here. And then we have a ZFN TALEN number 2, and that's going to match to this side, see that? And that's going to have a focal nucleus domain. And so once those come together, they're going to act like scissors essentially, very similar process, and snip these apart, okay? And that's why we get a nice symmetric break here, rather than the normal restriction enzyme breaks we've seen before. See that? Okay. Okay, is that making that more clear? Yeah. So the ZFN TALENs is the matching portion that we just looked at here. Okay, and they'll match a specific sequence. And then we'll have a focal nucleus domain attached to them. 2 of those added across are going to give you a break on your double-stranded helix. And we'll get a nice clean break between. The CRISPR-Cas9 system. So our CRISPR-Cas9 system is going to be modified to enable customized sequence-specific DNA cleavage. CRISPR actually stands, CRISPR, stands for clustered regular. Let me get some more water. Man, this was not the day to run out. Clustered regularly interspaced palindromic repeats, CRISPR. Okay, Have you ever heard of CRISPR, by the way? Yeah, it shows up in the news quite a bit. It's actually incredibly important, right? Yeah. Okay. So these are going to be found in genomes of many bacterial and archaeal species. Your Cas proteins, which are CRISPR-associated proteins. These are going to be proteins that cleave foreign DNA sequences as part of a prokaryotic immune system. The clusters of genes that encode the Cas proteins are near the CRISPRs. Why this is important is this is kind of our best hope to actually modify an adult human's genes. Okay? In case you're wondering. So this right here is your structure of your CRISPR guide RNA. We have a single-stranded RNA. By the way, single guide RNA, that's the sgRNA that we had, I guess I had actually mentioned before this. Sorry that you didn't get the wording for it. Single guide RNA, sgRNA right here. These are engineered single-stranded RNA. These are going to complement the desired target site. This is what this looks like. And so it has some sequence. This is then going to have these matching pairs right here. And this is the single guide RNA. The Cas9 protein, so the Cas9 is going to be a large nuclease, so it'll contain two lobes. The rec lobe, that is the binds to the duplex formed between the sgRNA and the target strand of DNA. The NUC lobe or nuc lobe, this contains the two nuclease domains that are going to be responsible for your DNA cleavage. So essentially you have a targeting and cleavage, right? Okay. The protospacer adjacent motif, which is the PAM. This is going to be a short DNA sequence that will be recognized by Cas9. The target DNA sequence complementary to the sgRNA is going to be adjacent to your PAM. The Cas9 complex will cleave both strands. And so this is actually what this looks like. Okay, so right here is your sgRNA. Everybody see that okay? Your red one? Okay. And then right here, blue and green, that's your DNA. Everybody see that okay? And then the sgRNA is going to match up to a portion of the DNA. See that there? Fantastic. Okay, now you're going to have two portions. You're going to have the nuc lobe and the rec lobe. Remember, rec lobe is going to be the binding between that to the complex, right? You can think of that as recognition, right? Nuc lobe is going to be the nuclease domains, which is what's named after. That will do the DNA cleavage, and that's your nuc lobe right there. Okay with that so far? There's your rec lobe. That's the recognition one. Okay, so here we have the target sequence right there. Right there's the PAM, okay? So right here we have the rec lobe, and it has right here the nuc lobe, and this is the Cas9 with the sgRNA complex. Again, the Cas9 being this part, the yellow and gray. The sgRNA is this part right here. Okay. Okay, so this is the portion that will actually match up. That's that in portion. Remember the in portion, repeated ins? That we can select whatever we want on it. Okay. Okay, see how they're even color-coded? So beautiful. Okay, now this is going to match up with a portion of your DNA. This is then going to bring that portion into the rec lobe. This is then going to allow the other portion into the nuc lobe. The nuc lobe is then going to be where you'll actually have the nuclease cleave that DNA. That DNA is actually going to be cleaved right here. Okay? Okay. So if the PAM sequence is present, the nuc lobe is going to cleave your target sequence. It'll cleave it somewhere in the target sequence, usually a little bit off the PAM. Okay? And then right here's your PAM. Now it does need the PAM to be present to do this cleavage. See, the PAM actually is right here. Okay. All right. And notice again, unlike the previous restriction enzymes we had talked about, this does not break it in sticky ends. Okay? This breaks it straight across. Okay? It cuts both strands. So the RNA interference is going to enable the disruption of the gene expression and presents new therapeutic opportunities. RNA interference, which is abbreviated RNAi, which is a great abbreviation. Somebody actually cared. The technique by which the introduction of a specific double-stranded RNA molecule can interfere with gene expression. The technique is called gene knockout, though you'll notice that this word gets reused quite a bit in various different contexts, okay? So you kind of have to be careful here. Dicer. This is going to be an enzyme that cleaves double-stranded DNA into fragments. These are called small interfering RNA. They're abbreviated siRNA, not to be confused with sgRNA, okay? Or just ssRNA. Anybody know what ssRNA would be? Single-stranded RNA? Yeah, like single-strand DNA. I know. We're getting a lot of terms and they're very similar. This is where the flip cards need to come in. The RNA is going to induce silencing complex, RISC, R-I-S-C, not to be confused with the 30 other RISC acronyms that are on Wikipedia. That was a bad choice, or at least it was a bad choice after the 10th, okay? I don't know which one came first. The assembly of the proteins that unwinds the RNA duplex, again, RNA-inducing silencing complex, unwinding RNA duplex, cleaves one RNA strand and cleaves the mRNA molecules containing complements of the uncleaved RNA strand. Okay, so this right here is the RNA interference mechanism, where we'll see this mechanism graphically instead of having to try to deal with them with the definitions. So right here we see the double-stranded RNA right there. Dicer is going to dice it up, right? So we've diced it, we've gotten a fragment. And so the next part is we're going to have RISC. It is going to bind this. We'll then have the cleaved passenger strand coming off. This is going to give us the mRNA binding to this. See that here we have one of the strands that are still there. We've cleaved the other one and released it. See that? Okay. So now we have this strand that's going to bind the mRNA. This will bind the mRNA strand because this now mRNA strand is in place of that previous other duplex strand. It will again get cleaved similar to the passenger strand before. Everybody see that okay? Okay. If you keep cleaving the mRNA strands, what's going to happen? You going to translate? Nope. And so we are what? Interfering with our RNA, aren't we? I know. Isn't that beautiful? Yeah, it actually is good terminology overall. The abbreviations are a bit rough, and there's a lot of parts to it. So it can be a bit of an issue, but each part makes a lot of sense, actually, right? Yeah. So foreign DNA can actually be introduced into plants, and this is something that's really wild about plants. So plants have a lot less care about introducing foreign DNA and new DNAs into their system. Part of that's because they can form essentially what we would think of as tumors, right? Plants don't have to move around with all that extra weight, do they? And so if we had that level of tumorage and excess baggage, essentially, we'd be weighed down to die, right? But a plant, oh well, I'll just grow over here now. So plants are kind of cool, but their DNA is so much more advanced than ours in some ways. The Agrobacterium tumefaciens, soil bacterium that infects plants and introduces foreign genes into plant cells. This is going to cause things like crown gall, which is a plant tumor at the site of the infection, but it can actually introduce entirely new genes, sometimes that are picked up from other plants. And so, for example, we've actually seen our GMO corn giving genes to other plants that are weeds next to it. And so now our herbicides don't work. Shit. They're beating us. The tumor-inducing plasmids. These are tumor-inducing plasmids, or TI plasmids. These are going to be responsible for the crown gall formation and the synthesis of amino acid derivatives called opins. Part of the TI plasmid is going to be incorporated into the plant genome. The TI plasmid derivatives can be used as vectors to deliver foreign genes into the plant cells. Again, just because genes are foreign does not mean that they are not beneficial, harmful. It just means foreign, okay? They could be beneficial, they could be harmful, depends. The TI vectors are going to be valuable tools for exploring genomes of plant cells and modifying plants to improve their agricultural value and crop yield. So this is electroporation. Electroporation is going to be the technique by which intense electric fields are going to be used to introduce foreign DNA into cells. I know, there's so many techniques to do this, right? In 20 years, this is probably going to be some of the most valuable stuff you picked up from school, by the way. So, honestly, keep your book 20 years from now, okay? Well, get the new book 20 years from now. It'll be better. The high electric fields are going to make membranes transiently permeable to large molecules such as plasmids. Does anybody know what transiently permeable means? I mean, sort of temporarily. Okay, so this is used to transfect corn and carrot cells with plasmid DNA that can include genes for things like resistance to herbicides, etc. It's basically just a cheaper, faster way to do these processes that we've already talked about. Okay, the other ones take a lot more work and a lot more money to do, right? In human cloning or human introduction or a vaccine used on a human, this would be probably a bad idea, okay? Because you'd get a lot of things out of this. But in plants, it doesn't really matter that much. A little error here and there isn't going to hurt anything. The cell wall here in the plasma membrane, and there we have the nucleus, right? And it has our DNA. Woo, plant. This is going to be we're going to digest the cell wall off first, and that will just leave us with the cell membrane, the plasma membrane, and the nucleus. So the next thing we're going to do is foreign DNAs then be added. We're then going to start transient electric pulses, basically electric pulses that move through the solution. Okay? And these transient electric pulses are actually going to cause that membrane to start having openings traveling across it. Okay? And these openings will be large enough for these foreign plasmid DNAs to insert themselves. I.e., another way of thinking about it is it makes that plasma membrane more mobile. Okay? And so these can insert through. We'll then regrow the cell wall. We'll incorporate foreign DNA into the genomic DNA right here. And now we have a viable plant cell with foreign DNA inserted. And this could be resistance to a herbicide. Okay? Or it could be a higher yield on wheat. Okay? All right. Anybody have any questions? No? Gene guns, because why not, right? Gene guns, or bombardment-mediated transformation. This can be a technique by which we take a 1 micrometer diameter tungsten pellet, we coat it with DNA, and we fire it at the target cells. Yeah. It's a gene gun. It's exactly what you thought. You want to do it now, don't you? Yeah. I know. I know. By the way, it's so funny because there's an old Civil War story that sounds so much like that, right? Where a guy was shot with a bullet. It took some sperm from him, hit a woman, and impregnated her. Yeah. You've never heard of that? Look it up sometime. They've tested it a number of times, and apparently very unlikely, but could happen. Which is very weird. Anyways, the techniques for introducing foreign DNA into plants allow for the development of genetically modified organisms. These are referred to as GMOs. You guys have probably all heard of GMOs before. Yeah, it's a big, like the non-GMO is very vocal in the United States. Interesting factoid, the non-GMO people are actually responsible for almost 1 billion deaths worldwide. Yep. They convinced Africa not to accept free crops that would have fixed their problems. Yeah. There's a really cool guy that's credited with, and there's a really cool guy that's considered the savior of 1 billion lives who did that for Africa, and then they convinced them that it would kill them. And anyways. So this can introduce the beneficial characteristics. It's things like ability to grow in poor soils, resistance to natural climatic variation, resistance to pests, fortified nutritional content. That sounds a lot like all the things that need to be done for our coming future. And we are the most anti-GMO we've ever been, which is interesting. It's like we were trying to kill ourselves. Maybe we are. The use of the GMOs, again, is highly controversial. The biggest thing you have to remember about GMOs is anytime we do something fast, we have a lot of danger, right? That's why in like your labs, I'm always doing reactions kind of slow, right? Slower than can be done. And that's because what do we call burning slow? Burning, right? What do we call burning fast? Explosion. Because it usually kills you, right? Okay. And so same thing with GMOs. Okay. GMOs are literally, peasants did GMOs 1,000 years ago, okay? Their GMO was, I select this thing because it breeds faster. I can give it to my landlord and he won't stick a sword up my ass. Okay? Yes. GMOs now are done really fast though, right? And so we don't have time to adapt to them, and we don't have time to see if they have any negative consequences before they become, dominant or everywhere. Okay? And so they are highly controversial. I'm not going to get into the entire discussion, but it is worth looking at if you're interested in it. Okay? It's actually not as one-sided as you might think. But the non-GMO people are fucking crazy. I'll just put that statement on record. Okay?